Two common types of cultures performed in a diagnostic lab are a throat swab and a urinalysis. Generally, the type of media we would like to use is blood agar, because most human-associated bacteria grow really well on blood agar. It provides a little bit of extra nutrients that allows some nice, robust growth. But sometimes we don't have enough blood agar for all of you, so then we'll use TSA, or what I like to say, blood agar. The urinalysis is going to require extra medium, so for urinalysis we'll use blood agar or TSA, and then we'll also like to use a McConkey or an EMB. These, remember, do pretty much the same thing. These media are selective for gram negatives, whereas blood agar is non-selective, right? So through using both blood agar and the McConkey/EMB, we can actually get a qualitative assessment as to the types of microbes growing. Are they going to be gram positive, gram negative, or do we have a mixture of both gram positives and gram negatives? So we can get that kind of stuff with our urinalysis, and also the ability, hopefully, to quantify, to get the right amounts. For a throat swab, it's very rare for the swab to go straight onto media. We are going to do that in the class, right? But generally, when you get your culture, we're going to use something like this. This is called transport medium, right? And this is a really nice, convenient way to get your samples, right? Because we have a nice labeling system here. I think now they even use stickers. When you open this up, you got yourself your nice sterile swab. This is kind of old, but it is a sterile swab. And then we have this gel-like, agar-like substance on the bottom here. And this does a couple of things. First of all, it provides a nice moist environment. It prevents bacteria from drying out, so the sample that you get is not lost over time as we transport it. It also has bacteriostatic ingredients in here to prevent microbes from growing. So once again, we're not going to overgrow whatever we get from your sample. So this, hopefully, keeps things as most representative of the sample as possible. All right, so let's show you how to do a throat swab. So in your book, there's a diagram as to how to grab it, right? But imagine this is the mouth. You just take your swab and then all the way to the back. You want to make sure it comes from the throat, right? Don't touch the tongue. Don't touch the roof of the mouth. Don't touch the sides of the mouth. Don't touch the little dangly thing. All the way to the back. Get yourself your nice little sample. And then now we have our swab, right? Once again, normally we would stick this into our transport medium, right? But we are going to go straight onto agar. So simply take your swab, and then you're going to make a Z. Zip. As I'm turning, I'm going to make a Z like that. Okay? So just make a Z, and then make sure you rotate your swab as you do it. This is biohazard, so I'm going to throw this away. And then I'm going to take my swab, my loop, nice and sterile. And then we're going to streak for isolation. And you can see the diagram in your book. We actually are going to cheap out and split this in half, right? So imagine this imaginary line down the middle. And all I'm going to do is go like this, and then just streak it out like that. We are not going to try to quantify. We're not going to try to do anything. Just see what types of microbes are living in the back of our throat. And we'll have two conditions, which we'll talk about in class. But that's throat swab. Pretty simple, right? So I'll push that out of the way. For urinalysis, again, we will have two media, a blood agar, right? Probably TSA, and then McConkey or EMB. We will have some urine for you to goof around with, but today I'm just going to use water. And ideally, we would have a calibrated loop, which picks up a specific amount of liquid. I think it's 10 microliters that the calibrated loop normally is. But again, we don't have enough calibrated loops, so we'll just use our regular loop, and we'll call this a calibrated loop. Okay? So two ways to do this, and I'll just show you both ways, and maybe I will let you choose. But when you do it, make sure that you use the same technique for both plates, because we do want some sort of consistency. All right, so the spreader method, this is the method that I prefer, is simply like this, right? You will just get your, oops, that was still hot. Just get your drop on your loop, and that's our simulated urine. For me today, it's water for demo purposes. Lift the lid, and then just place that drop right in that center. And our calibrated loop will deliver exactly 10 microliters, right? And it has to deliver a specific volume because we do want to quantify, right? And in order to be able to quantify, we need to know exactly how much is in there. Move this out of the way so it doesn't burn. Our spreader, remember our friend from the past, gentle, right? Not tap too hard. And then we're going to burn that ethanol off. And we're going to do it one more time. Nope, I still see a little flame, so I am going to do it a third time, making sure that there's no flame. Lid near our flame, and then I like to do this, touch, 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 touch. And then remember, counterclockwise turn, clockwise with my spreader. 10 microliters is a very small amount, so this will probably take about 10 to 15 seconds. And we are done. Close the lid, put this away. That's the spreader method. The other method that we can do, I'm going to bring my fake urine back. I'm going to sterilize our loop, is using a loop. So that you would do if you don't have a spreader. And sometimes when people break our spreaders, we don't have spreaders. So if you are instructed to do the loop method, it starts the same, of course. Grab our urine sample, and this time, put it down, we are going to go top to bottom. I'll show you that. I guess bottom to top, just like that. And that's our inoculation. Now we're going to sterilize our loop, right? So now we have placed 10 microliters of our urine sample onto the plate. Let that loop cool. And then now I'm going to use my loop to spread the urine sample, very much like I would with a swab, right? So this was where my line was, up and down. So I'm going to go like this, turn it on its side, and then just back and forth one time, top to bottom. And then hopefully that'll have it nicely spread out. Hopefully we don't have too much bacteria. Hopefully we can quantify the amount of microbes in that sample. And that's it. That's our throat swab, our quantitative urinalysis. Through this analysis, we can then try to figure out, A, how much bacteria is present in the urine, right? And there's a scale that's in your book that'll let you know whether you have no significant growth, whether you have infection or an acute infection. And then hopefully we can also get a qualitative analysis as to the general types of microbe present.